Thank you for that introduction. So as you mentioned, my name is Chris Hill. I'm working out of the National Institute of Health in the United States, as well as the University of Pennsylvania. And so today I'll be presenting on this proceedings talk titled ChromDL, a next generation regulatory DNA classifier. So just a high-level problem statement of the issue that we are working on as part of this model. So there's a good number of large sequencing data sets that have been made available over the past decade or so that have led to a number of algorithms that have been developed for the prediction of chromatin features and regulatory regions, specifically in human genomes and human DNA. And as time has gone on, the improvement of these different machine learning libraries, better GPUs with more memory, and more sophisticated optimization algorithms that can bring training time down from weeks or days into hours or minutes at a time, have allowed many different types of architectures to be created and tested much more rapidly. And so that's exactly what we sought out to do. We wanted to combine many different architectures, including these convolutional neural networks, these recurrent neural networks, and a whole bunch of different normalization, pooling, et cetera, layers, and see how we could perform on these same classification tasks now that we can test these models more quickly. So just some history on the search space. So the pioneering method for this DNA sequence analysis that we were focused on here is out of this group that created this deep convolutional neural network architecture back in 2015. And so this group took 1,000 base pair input genome sequence that was compiled from the ENCODE and roadmap epigenetics data sets, fed this through a 3-layer convolutional neural network for the prediction of transcription factor binding sites, DNase I hypersensitive sites, and histone marks in the human genome. The group then went on to do some functional invariant predictions with this output, but we are most focused on the deep convolutional neural network and these regulatory feature targets. So just in investigating this architecture, because we'll do some comparisons between our model and sort of the history, this is a traditional 3-layer convolutional neural network. We have this one hot encoded inputted DNA sequence, 3 layers of increasing kernel size with radioactivation, dropout, and pooling. We have this dense layer before feeding into this 919 output feature target. So we have the transcription factor binding sites, the DNase I hypersensitive sites, and the histone marks as a part of this. So a very traditional architecture. Sort of the second generation of this came out a year later in the form of DanQ and DanQ JASPAR. So the architecture on the left, some similarities as you've seen with the previous deep C architecture. We have the same data set, same one hot encoded input, a similar convolutional neural network, but we also have this inclusion of a bidirectional long short-term memory layer before the feeding into the dense layer and the output layer. And so this group proved that this combination of this convolutional neural network and this bidirectional LSTM could lead to an improved output in the form of better area under the receiver operating characteristic curve. And so they provide a series of plots like the one on the right, where just you have the same features from the same data set being plotted, and their model performed better with this area under the curve metric. And so with that history in mind, we wanted to take this a step further, see what we could do with all these different recurrent neural network layers and the convolutional neural networks, and combine many different architectures in intricate ways that have not been attempted just because of time constraints, memory constraints, et cetera. And so we combined all sorts of CNNs, RNNs, gated recurrent units, LSTMs, dense layers, pooling layers in very strategic ways, and fed them through our GPUs. And we could, because of the advances in the optimization algorithms, these models will converge to their predictions much more quickly in fewer training iterations. And so we could more quickly parse out those models that appeared to be promising from a few iterations of training. So that's what we did. And we would parse out the promising models and we run them for much longer, for 100 training iterations, to try to maximize this area under the curve scoring. After which we did some analogous layer replacement. So we would take convolutional neural networks that were standard, replace them with separable ones. We would take unidirectional layers, replace them with bidirectional layers. We would do sort of swap outs of RNN types, so LSTMs for GRUs for simple RNNs, et cetera. And this final ChromDL model was the highest scoring area under the receiver operating characteristic metric model that we extracted through these investigations. So here's the architecture of our final model through all these investigations. So again, I'll highlight a few similarities with DeepC and DanQ as well as a few differences. Again, I want to highlight the same data set is being used for the input with the one hot encoded sequence. The same output 919 feature map target is being utilized. A few notable differences here, the first of which is this bidirectional gated recurrent unit input sequence. So the DNA sequence is immediately fed into this bidirectional GRU layer as opposed to the more traditional convolutional layer, which we found improved our model in these performance metrics. We then have a layer of separable convolution where we first have a depth-wise convolution that acts on the channels separately, and then a point-wise convolution that mixes the channels together, followed by a standard convolutional layer, and then two sequential bidirectional LSTM layers. And this bidirectional layer just means it reads sequence front to back and back to front simultaneously and shares weight between them. We also have the absence of a dense layer between this bidirectional LSTM and this output feature target, which we also found improved performance. So just in sort of the tradition of the predecessors, we were looking at this receiver operating characteristic curves as our primary metric. Each of these gray lines here is one of the experiments of the 919 targets. So in particular for transcription factor binding sites, we see very high area under this ROC curve scoring, where we're trying to maximize those true positive rates and minimize the false positive rates. And then down below, just a table of the proportion of peaks in which our model is outperforming these comparison models. So in particular for the transcription factor binding sites and the DNase I hypersensitive sites, we have a very high outperformance of ChromDL compared to these three models in the 90s or the high 90s for the DNase I hypersensitive sites. Just to sort of affirm this, we performed a bootstrapping analysis to try to estimate the true mean area under the curve for the ROC and also the precision recall curve. And we found when we did this, we obtained the significant increases in the estimated true area under these curves compared to these comparison models, and the following median area under the curve scores for these two metrics. So great, we have these area under the curve scores, but we want something else besides these metrics to base this improvement of a model off of. So the next thing we did was we looked at the low signal transcription factor cheap-seq peaks that were included in our data set. So as in all machine learning training models, we have our training set, our validation set, and our test set, which is not been seen by the model, so we can feed it through and have it make new predictions on the inputs. And so we're interested in particular in low signal strength transcription factor cheap-seq peaks that were provided in the data set. So these signal strengths came with the data. And we wanted to see if ChromDL did a better job at predicting these low signal peaks in comparison to the predecessor models. And we found that, in fact, across all of these groups, there's a significant increase in the mean proportion in which they're predicted correctly in each of these comparison models. And just to clarify here, so each of the four models is fed each of the cheap-seq peaks separately, and all of them are expected to be predicted correctly because they are, in fact, all measured cheap-seq peaks. But this graph just captures those that were correctly identified as positive peaks. And so the high signal strength ones perhaps predictively were identified most easily as compared to the low signal ones. So as a step in validation, we wanted to see, we have this ChromDL model now with our pre-trained weights. We wanted to see what we could do with this. So the first step was making a secondary enhancer classifier that would take the output from ChromDL and feed it through a second three-stage CNN before predicting enhancer regions. And so this architecture, this TreadNet architecture, was made by a member of our laboratory and is similar to kind of the deep sea picture that I showed earlier, more of a traditional three-step CNN with this dense layer leading to one enhancer score. And so we trained this two-stage model across all available cell lines that we had in the data set for 100 training iterations across different enhancer definitions based on overlapping histone marks, et cetera. More details of that are in the manuscript, but to get this one enhancer score. And we were able to achieve pretty comparable area under the receiver operating characteristic scores for this enhancer prediction task, which we found encouraging. But we were also interested in the sensitivity of these enhancer regions to point mutations. Could this model still hold up when we were changing the sequence that's being fed in? So in investigating this, we looked at reporter assay quantitative trait loci, so just point mutations that are identified that have a significant impact on regulatory regions in the genome. And so we were curious to see if we have our mutation, if we feed in a wild type allele and then this mutation allele, if our model, this two-stage model, will be affected if the score will be impacted correctly as it should by these mutations. So that's exactly what we did. We took this 1,000 base pair input, we fed it through both of our pre-trained models. We had two versions of each sequence, one with this mutation and one with the wild type allele. And we found that we could calculate a delta score, so just the difference in the enhancer scores from these two sequences fed through the two-stage model. And what we found is that the highest delta score ROC QTLs, so those that had the greatest effect on the two-phase enhancer score, had the largest percentage in our defined enhancer regions, which is exactly what we're looking for, right? If they're disrupting enhancer sequences, either by activation or deactivation, we would expect to find that in the greatest proportion in our enhancer regions. And indeed we did. So we believe this validated our model. One final investigation we did as a part of this was meme sheet motif detection. So we wanted to see if ChromDL did a better job at parsing out the transcription factor motifs as compared to these other models. And so in analogous to the low signal transcription factor investigation, we fed the peaks that were in the testing data set through each of the four models separately. We isolated only of those that were positive for each of the four models individually, and we ran the meme sheet motif detection tool on each of these. So we wanted to see if some models were better at parsing out these motifs than others in more of an experimental type investigation as opposed to just the rote machine learning metrics of the area under the curve. So we have these published PBMs, these position weight matrices, or excuse me, protein binding microarrays with these PWMs, these position weight matrices, as well as CELEX database of PWMs, which are just sort of the comparison of what we are hoping to parse out in these motif investigations. And so we have 306 of our 690 TF CheapSeq peaks in the data set we could utilize for this investigation. And we found that on average, ChromDL had the highest similarity score using the TomTom alignment tool in comparison to DeepC, DanQ, and DanQ JASPAR with these motifs. This figure just wants to illustrate that it's not anecdotal, but it's also not the case that in every single example, ChromDL outperforms the other three comparison models. So in this first column on the left, for each row we have, so the top row is the comparison model, then each row is the model itself. So second row is ChromDL, third row DeepC, DanQ, DanQ JASPAR, and each column is a different PWM that we're looking at. So this first column, this PBM June, we find that ChromDL picks up this GC dimer much better than DeepC and DanQ models, which is a very encouraging result. But taken alone, we don't want it to misrepresent that it always, outperforms these other models because they are still useful. So this second column then, this CELEX HSF1, we have ChromDL still performing the best, most similar in the alignment using TomTom to this top comparison motif. But we have DeepC here that also does a pretty good job and captures this GAA sequence that DanQ and DanQ JASPAR did not, despite the fact that DanQ has a higher area under the curve for, on median metrics. And then this final example on this right column is just to show that there's plenty of examples out there where all of the models do a great job of parsing out the transcription factor motifs. So we wanted to include this to highlight the usefulness of ChromDL and the excitement that we have for it in this transcription factor motif detection, but also the usefulness of previous models and sort of using them all in tandem and just keeping them in mind in investigations. So sort of just a discussion of future directions. So we believe this model really highlights the benefits of these RNN layers in addition to the CNNs. So taking a step further than DanQ did of just including the bidirectional LSTM layer, we also have this input bidirectional GRU layer, which has not been used in previous iterations in this two-layer back-to-back of bidirectional LSTMs. We also have the improvement accuracy in the low affinity transcription factor binding, the potential of this transcription factor binding motif identification performing better than these comparison models, and on average across our entire suite, having a higher alignment score. And just the usefulness it could have as a secondary classifier in downstream tasks of prediction enhancer sequences, as an example, that are still sensitive to the regulatory region mutations. So we believe that our model provides enough context for this downstream models to perform effectively. Future directions, could look at lowering the training time, which will inevitably come with better GPUs, better ML libraries as time goes on, initializing the models with motifs similar to how DanQ JASPAR did, and then just continuing to include more data sets as they become available. But as it stands, we believe it's an accurate regulatory classifier. We are only using DNA as an input, and as a result, because we're using the same data set, we believe we can compare it fairly with DanQ and DeepC. And we believe it's a very useful tool for the development of new technologies and further investigations in epigenetics. So just to acknowledge my team that I worked with at the NIH under the Ultrarenko Research Group, as well as the support that I received from the intramural research program that funded this work when I was there. And then the GitHub repo is just the open source ChromDL. This is available. We're working on turning it into a Python package. And then the QR code is just the manuscript if you have not had a chance to read it. So thank you.
Thank you very much for your talk. So it was very interesting to see that you included this gated current evidence just after one had going. So I've never seen it before, or somebody did. So can you maybe have a slide of ablation study to show that indeed it improves your performance? No?
I do. It's not included in this slide, but we did a series of individual and dropout layer performance and found that the inclusion of this did in fact increase the performance.
What is the intuition for the audience? What is the intuition? What does it capture? What is seen as not captured?
Sure. Difficult question to answer. Not entirely sure because it is unorthodox, as you mentioned. So we didn't do an investigation to the sort of the interpretability of the layers so much as the performance output. But you're right. We're not certain why what it captures spatially or sequentially out of the data as opposed to the spatial convolutional layers. So, sorry, I don't have a more satisfactory answer than that.
The question from the back. Sorry, the back.
Thanks. Very interesting talk. So I hate to say it for the people in the audience, but 2015 was almost a decade ago.
Yeah.
You say that ChromDL is the next generation sequence classifier. Have you tried comparing against the current generation of sequence classifiers? Like there are two other methods after DeepC from that lab.
Sure.
Have you tried looking at those?
We did. When this investigation started, we kept the focus on DeepC because of its usefulness just across our lab and neighboring labs, and we kind of left the focus on that. But you're right, as time has gone on, even as this work has progressed, there have been more models that have been produced. And this is just a tool to add into the data set or into just the available library of tools to utilize. We should absolutely look into more investigations and how they perform with these current tools. But this was more of a next generation as opposed to the initial data set and this initial investigation.
Yes, thank you.
Maria had a question here.
I have two questions, but they're really short.
Okay.
One, did you look at like the standard kind of things like dilated convolutions and skip connections? I just can't remember if that was on the slide because it didn't make it into your final model. And the other question is you mentioned advances in optimization. What specifically?
Oh, sure.
Being that?
So the first question, we did not look at skip connections. We looked at attention for a short period of time, but it added a great deal of complexity, so we ultimately left it out in favor of just the combined CNN and RNN layers. I'm so sorry, I forgot your second question.
What are the optimization advances?
Oh, sure. So the algorithms itself is what I was referring to. So the early generation used as SGD algorithms for just the optimization, which take a much longer time to train and much more training epochs, iterations. And our model utilizes the Atom optimizer, which just converges much more quickly in comparison to these older algorithms. That was my reference to.
Okay. Final question here will come from Naoki Osato online. Do you have a plan to make databases of predicted regulatory motifs though?
It's a great idea. We haven't so far. I think the next steps will be just making it as a Python package and a consumable to utilize, but absolutely the resulting motif libraries should definitely be something we look at as well. So,
thank you.